No, I still have this.
Oh yeah, actually, yeah.
Oh, you have it too?
Yeah,
Okay.
it's natural repellent.
Oh, I have it. Yeah.
Okay,
Okay, great.
yeah, let's put some. On my hand especially,
Yeah.
because there are many ticks.
You want some?
No, I'm fine. I don't need that.
No? Are you sure?
Yeah, really, no.
Okay, that's fine.
Let's go.
It is common for researchers to use body products such as sunblock or insect repellents during fieldwork to avoid getting sunburn or bitten by mosquitoes or ticks. Several diseases can be transmitted by these insects. Lyme, transmitted by ticks, is the most common one in the Netherlands. The number of patients diagnosed with Lyme's disease each year has increased since 1990, reaching 25,000 new cases a year by 2015. There are different types of insect repellents available on the market. Deet is the most popular repellent among these. However, natural repellents have become very popular in recent years. The repellents are intended to drive off mosquitoes and other biting insects, but may have the same effect on other groups of arthropods. Therefore, when researchers wear insect repellents and or sunblock, it is possible that these chemicals could be transmitted to traps in the field, altering the results and the validity of the study. We test this question with a study on ground beetles. In 1981, a researcher argued that ground beetles are repelled by Deet. Knowledge about behaviour of beetles exposed to Deet and natural repellent in the field is scarce, and studies about the effect of sunblock are absent. Ground beetles are a family of mostly carnivorous beetles. There are more than 40,000 species worldwide. Researchers are interested in studying them because they are good indicators of the biodiversity of other arthropod species. On Google Scholar there is currently 265,000 books or articles on ground beetles, and it is likely that researchers in a number of these studies use body products which may have influenced their results. Repellents applied to the skin or clothes produce a vapour layer that has an offensive odour and smell for insects, modifying their behaviour. It is known that mosquitoes are repelled by Deet because it jams their smell receptors. Citrodiol, used in natural repellents, works the same way. Ground beetles' density is affected by several factors. First off, important factors consist of the predation rate and prey catching rate. On the one hand, their predation rate depends on the predator abundance as well as the ground beetle's ability to spot predators either by sight or by chemical cues. On the other hand, their prey catch rate is modified by the prey abundance and their ability to find prey either by chemical cues or by sight. We expect the placement of pitfalls contaminated with repellent to affect the ability to find prey and spot predators by chemical cues. This increases predation rate and lowers prey catching rate, lowering the beetle density. Furthermore, other factors may also influence beetle density, namely humidity, temperature, season, life history, vegetation type, and the thickness of the litter layer. We tested this hypothesis by setting up a field and a lab experiment. The research question we tried to answer is, can the presence of body products, namely Deet, natural repellent, and sunblock, decrease the catching rate of beetles? In the field, the treatments Deet, natural repellent, and sunblock are applied around pitfalls. This is done in order to imitate the possible contamination that a researcher could cause when applying those products during the field work. The presence of beetles caught in the treated pitfalls will be compared against the control pitfalls. We predict that Deet and natural repellent will dissuade beetles to approach the pitfalls where the treatment is spread. Therefore, the presence of beetles in Deet and natural repellent traps will be lower than the control. Because there are not any previous studies about the effect of sunblock on arthropods, we cannot predict the repellency or attraction of beetles to sunblock. For this reason, the prediction of the sunblock effect is comparable to the control. To reach the different sites, we had to cross a lot of undergrowth forest, which was hard work and exposed us to an unpleasant amount of ticks. dug in pitfalls and administered the different treatments. Natural repellent and DEET were applied, with 3 sprays around the cup, and sunblock was applied by putting 8 drops of sunblock on a glove and touching the ground around the cup. After applying the treatments, each pitfall was covered with a roof to avoid predation, water inflow, and capture of vertebrates. Each site consists of 3 treatments and the control group. The treatments used are DEET, natural repellent, and sunblock. The different treatments and the control are situated 5 meters from each other, because it is expected that the effect range of the treatments is less than this distance. Each treatment and control consists of 3 pitfalls, situated 2 meters apart. We used a block design to control the large spatial variation in beetle abundance. On the first day, 8 sites were placed in a straight line in the eastern direction, at least 50 meters apart from each other to prevent the treatments in the blocks from influencing each other. On the other days, this line was moved 50 meters in the northern direction. We changed its location on a daily basis to avoid pseudo-replication by the influence of past treatments in the same area. The different blocks were placed in a grid in the Oostereng forest between Bennekom and Renkum. After 6 hours, we picked up the cups, and the number of beetles and other arthropods found inside them was noted down. The number of beetles caught in the pitfalls was transformed to presence-absence data to be analysed with a logistic regression. The results show that the treatments applied do not have a significant influence on beetle presence. However, we observed some differences between the different treatments. Apart from beetles, other species were caught with the pitfalls as well. Many individuals of other groups of arthropods were also found in this experiment. Mainly, we found spiders, woodlice, centipedes, and springtails. In this case, a significant effect of the treatment in arthropod presence was found. The treatments that were significantly different from the control are DEET and sunblock. Finally, in order to test the community response to the treatment supplied, all the arthropods that fell into our pitfalls were analysed in another logistic regression. The results showed that the treatments had significant effect on the presence of arthropods. Again, the treatments that were significantly different from the control were DEET and sunblock. Several covariates which were expected to influence the results were measured. These variables are time in the field, shade level, thickness of the litter layer, distance from the road, and vegetation type. A principal component analysis was done to gain more insights in the effect of the treatments on the different species composition. It shows that control and natural repellents are similar, whereas sunblock and DEET are also similar, but the 2 groups are different from each other. All the species point towards control and natural repellent, indicating that sunblock and DEET have repellent effect. Woodlice and spiders were found to be the most abundant arthropods in the area. Finally, the environmental variables have little importance in the effect of the treatments. A second principal component analysis was carried out to assess the effect of the vegetation composition on the presence of arthropods. Results suggest that a great part of the variation is already explained by the treatment, and that the vegetation composition has little explanatory power. The field data showed a significant effect of the treatment on the overall arthropods. The beetles alone didn't significantly react to the treatment, but showed the same pattern as the other arthropods. For this reason, we decided to carry out another experiment in the lab just on ground beetles to test the repellent effect of the treatments under controlled conditions. In the fieldwork, we gathered ground beetles of 2 species to test in the lab. The species which were caught were the most used, which are Nebria brevicolis and Carabus violaceus. It is not clear if sunblock would repel or attract ground beetles because of the lack of research on this topic. Therefore, we expect that sunblock would generate the same data as the control. DEET has been tested before on the repellent effect on beetles, and so we predict that DEET will repel the ground beetles. Natural repellent has been created to repel mosquitoes and other insects to mimic the effect of DEET. Therefore, we expect that natural repellent will also repel the ground beetles. The lab experiment was carried out to test the repellent effect of the 3 substances, sunblock, natural repellent, and DEET, under controlled conditions. The experiment setup consists of 12 containers. For each treatment, we use 4 of them. Each container consists of 2 PVC tubes connected by a central T-shaped pipe fitting. The treatment is situated on one side of the container and a positive control on the other. The inner end of the tube was partly covered by a piece of cardboard with an opening to allow the beetles to pass through. During 6 days in the morning and afternoon, the experiment was run. This led to a total of 44 replications per treatment. The experimental units are ready to use. Therefore, the experiment can start. In order to avoid the possible residues between replications, aluminium foil was placed with a piece of paper on top of it, and for each replication, these are replaced with new ones. The control consists of spraying 2 times with water on top of the paper. The treatments, natural repellent and Deet, are also sprayed 2 times on the dry paper. The last treatment is Sunblock, in which 5 drops are placed on the paper and then spread out with the finger. On all treatments and controls, a litter layer was allocated to make sure the beetle moves to one of the 2 sides of the tube. The ground beetle had 3 hours to decide in which compartment it resides. After 3 hours, the allocation of each beetle is checked by removing the cap in the middle and by putting a piece of cardboard in the tube to make sure the beetle stays in the same compartment. The result of the experiment was either the beetle on the treatment side or the control side. As shown before, each of these experiments generates binary data because the beetles had 2 choices. Each treatment was analysed separately to look for significant preference for treatment or control. The data has been analysed with a binary logistic regression analysis. It was expected that the amount of beetles found in Sunblock would be the same as in control. However, the statistical analysis showed that Sunblock had a significant repellent effect on ground beetles during the lab experiment. The same lab experiment was performed for the natural repellent. The prediction of the natural repellent was that the ground beetles would be repelled by the substance. Analysis indeed showed a significant repellent effect on the ground beetle. For Deet, it was expected that ground beetles would be repelled by the substance. The analysis of the lab experiment with Deet showed a significant repellent effect. All 3 experiments showed that the time of day, morning or afternoon, and species do not have a significant influence on the data. Therefore, these variables do not affect the results we found. The lab experiment showed that all the different treatments have a repellent effect on beetles under control conditions. Deet, natural repellent and Sunblock are widely used during fieldwork by researchers. But is this a recommendable practice? In the field experiment, we have found that Deet and Sunblock can alter the catching probability of arthropods. Overall, we found a significant effect of the treatment on a wide group of arthropods, but we did not find any significant effect of the treatments on the probability of catching beetles. They showed the same trend as the other arthropods, though there was no statistical data to prove the difference between the groups. In the lab experiment, we can agree with the argumentation in the literature that Deet repels beetles. However, our research suggests that natural repellent and Sunblock have a similar effect. The difference with the field results is that in the lab the natural repellent had a significant effect, whereas in the field it did not. We expect this is the case because in the field the pitfall was out for 6 hours, whereas in the lab the experiment only ran for 3 hours. The natural repellent probably does not repel for many hours, making the effect in the lab stronger than in the field. Whilst we find significant effects of several treatment groups, it is possible that the body products do not significantly affect results in practice, as in this study we use quantities that would normally not appear around pitfalls when handling it with contaminated hands. Therefore, we suggest to expand our field experiment to test different quantities of contamination in order to show if there is still an effect on beetles with lower quantities of body products. Always be aware of the kind of products that you use or wear, because it could alter your research and introduce bias.